Dunnit, DNA fingerprinting and forensics. Imagine you're a fresh, young crime scene investigator, or CSI for short, and you've just come across your first crime scene. You found traces of blood from the probable criminal among the evidence. Now, if we were living way back in the 1800s, your boss would probably tell you to get a mop. Good thing we live in the 21st century. To help find the criminal, we're going to use something called DNA profiling. DNA profiling, also called DNA fingerprinting, involves analyzing DNA in order to identify someone. You might recognize this process if you've ever seen the Maury show, as DNA profiling can be used for proof of paternity. Maybe you've heard of all those ancestry DNA kits that everyone has been talking about. It's all DNA profiling. The technique was discovered in 1985 by Dr. Alec Jeffreys at the University of Leicester when he saw that different DNA samples showed different barcodes when separated. But how does it work? After collecting a sample, the DNA is separated from the cells, copied, and then run through a process called gel electrophoresis, which separates DNA based on size. Imagine the process of gel electrophoresis as separating Russian nesting dolls. Now we have the specific barcode that separates that individual from everyone else. This barcode can be used in two ways, either by comparing it to another suspect's DNA and looking for a match, or by running it through a DNA database, like CODIS. If you've ever watched a crime TV show, you might recognize the term. It's the federal database of every convicted criminal's DNA and stands for Combined DNA Index System. Now, this technology seems amazing, but just like people, this process isn't perfect. It can be contaminated if the wrong DNA gets into the process, as well as by laboratory error. But don't lose hope. DNA fingerprinting is still a wonderful process that helps investigations every day.